Hello and thanks for joining us today for our latest Technology Networks webinar, Unlocking Comprehensive Genomes for Large-Scale Projects. I'm Dr. Karen Stewart, Senior Scientific Specialist for Technology Networks, and I'm here to moderate the event. I'm pleased to have Cora Vasher, Greg Elgar and Brynja Sigurpelsdottir joining us today as your presenters. A very warm welcome to you all. Cora is the Market Segment Associate Director for Human Genetics at Oxford Nanopore Technologies. She's passionate about genomics, in particular how genomics can help decipher and alleviate the burden of neurological diseases, from neurodevelopmental to late-onset neurodegenerative disorders. She came to the UK for a postdoctoral position on the genetics of Huntington's disease in Cambridge, and subsequently moved to commercial organisations. Greg has 40 years' experience in molecular biology, including 30 years in genomics at principal investigator level. He's co-authored over 150 peer-reviewed publications and played lead roles in a number of international genome sequencing projects. Greg's academic career includes positions at the Sanger campus, QMUL, MRC NIMR, and the Crick Institute. He established an R&D lab at Genomics England in 2018, establishing Nanopore whole genome sequencing with the aim of clinical implementation in both cancer and rare disease. Greg also coordinated epigenetic, transcriptomic, proteomic and metabolomic programs in research in rare disease. Brynja is a research associate at deCODE Genetics and working on her PhD from Reykjavik University. She did her undergraduate studies in biomedical engineering at Reykjavik University and later studied bioinformatics at ETH Zurich. For the past 5 years, she's been working with long-read sequencing methylation data. Following the webinar, we will have a Q&A session, and we welcome any questions that you may have. You can submit questions for the Q&A session at any time during the presentation. To ask a question, you should enter your question into the box on the right-hand side of your screen and click Send, and we will answer as many as possible during the time available. For any questions we don't have time to get to, we'll make sure you're contacted offline with an answer. If you experience any technical issues, please click the chat icon in the bottom right corner to request support. And please remember you can ask questions at any time, even if you're watching this webinar on demand. So, without further ado, I'll now hand over to Cora.
I'm going to give a very short introduction on showing that we're really coming towards the era of complete genome. And we will show with our external speaker that we can do that at scale. So you may all have seen now that the telomere to telomere has been completed in 2022. That's really radically improving what we are knowing today. Nanopore ultra long reads have proven very necessary in completing a T2T genome. It's now moving, the genome of reference are now moving from T2T to more pan-genome to have a better representation of genomic landscape across diverse human population, and having phase genome assembly, really realizing an increase of gen and mutation conversion to phenotype. In the word of Michael Atwaters, the use of a reference genome that is more representative of the population of a given patient will be more accurately linked genomic variant to the disease. And even the Y chromosome, pesky little white chromosome, is now fully sequence telomere to telomere. So we're really moving towards a true genome reference. So there's been lots of presentation here. Still, there's a lot of exome going on. There's still a lot of short-read whole genome going on. And then some new small variants, some SNP, some indel that are being associated. If any of you went to the Genomic of Africa this morning, in non-European population, there's still a lot of discovery to be made. But what short-read doesn't give you, it's information about larger and much more complex variant, structural variant, copy number, repeat, etc. What sequencing with it bisulfite doesn't give you, it's epigenetic, and it's really important. Methylation is really highly involved in complex disease and in cancer, and we'll have a presentation focusing on that. And it's only partially visible through modified chemistry, whether you're going to do bisulfite and epic or bisulfite and short-read. It's also very algorithmic, heavy on interpretation. And last but not least, even if you're focusing on exome, you're going to miss by short-read exome a lot of the dark genome. So it can be coding, but it's still not captured because either the region is not amenable for sequencing band synthesis because it's too GC rich, or the alignment is too complicated because the region is too duplicated and too repeat heavy. And so we really need a reference genome that is going to be the central element for everything, clinical, comparative, and population genomic analysis. So what does it mean in terms of application? And what it is that we can help you in terms of your application? I'm going to start from the end bit there. Because I've just presented and explained about reference genome, we can really do telomere to telomere assembly. And I do have a slide on that, but we are the only platform that can provide a true telomere to telomere assembly without any gap, with a new bundle of reagent. I don't expect a lot of people to do full telomere to telomere, but I will also have a slide to show the value of doing ultra long read for assembly level of reference genome with very long contigs and maybe just 1 or 2% gap. And then we move towards what you guys are very now comfortable with, either doing a standard whole genome sequence that you would use maybe for translational or clinical research, where you will be able to capture everything, SNPs, indel, methylation, SVs, and the long read presentation yesterday really show that we can do that. We can do that very, very well for rare disease. But if you have a lot of sample and you might want to put more sample per flow cell, so 30-fold, we recommend 1 sample per PromethION flow cell. But if you have a lot of sample, if you want to study inter-individuality over really deep characterization of specific variant, because you're looking at rare disease, we now can support multiple sample per flow cell, and you'll still be able to capture SNPs, capture very well structural variant and methylation at a slightly lower pass. So 2 sample per flow cell, you get 15, 20-fold coverage. And obviously, we do have a solution for genotyping. I don't mean arrays, I mean targeted sequencing. There was an excellent workshop with Asuragen really showing that we can really help with targeted solution. We're also working with other company. If you talk to Twist, they have a new poster that we're developing with them. Sorry. So I'm just going to go very quickly around what you get, high quality from a human genome in a single flow cell. And again, if you went to the long read presentations yesterday, I'm going to present my favorite feature in the order I like them better. And it's not the same. So I think for me, I'm a methylation person. Modification calling at 99.7 accuracy, you do get methylation and hydroxymethylation within your data. You don't need to tweak it. You just need to tell the instrument, yes, I am interested in getting the methylation signal. There is no treatment. There's no additional work other than the bioinformatics. Obviously, long read is really associated with structural variant. At that point, I want to remind everybody that we will read any read length. So whatever you put through the pore will read. Obviously, when we're talking population, genetics, and rare disease, we are more interested in the long read. But if your application requires short read, we absolutely can support you with that. And phasing. We can generate megabase scale phasing. And, we can do SNP calling as well as short read, as well as everybody else. So we do have end-to-end protocol. Again, with 1 PromethION flow cell, you can get a whole genome that does give you all that accuracy. And we now have automated analysis on our Happy2Me platform. We had a number of demos on the booth. I'm going to have three, talking about 3 points that are specific. It's all very good generating new long-read data set for rare disease, but what do you do with the new variant that you identify, especially if they're structural or they see evidence? How are you going to categorize and prioritize those new variants if you don't know their frequency in the population? So we're working with the 1,000 Genome Project and created a consortium to really expand our understanding of the human genetic variation. The data are available to everybody. I think Danny's Miller is in the room. If you do want to access the data, please come to him and he'll enable you to access the data. At the moment, there's about 200 samples done. We're planning to do a lot more with them, about 800. And it's really generating a catalog of structural variation to filter and enable variant prioritization. It will definitely expand our understanding of variation, especially in difficult to analyze region. And the group is also evaluating which variation need to be looked more into. We had our own conference last week, London Calling. We had about 1,000 people on site, about 3,500 people online, and we announced a number of new product and development. There's just one I wanted to point to here, just because it's very relevant to population genomics and at scale. It's that we can now have telomere to telomere bundle, which will include ultra-long read, PORC for chromatin conformation, and an assembly polishing part of the kit to really enable the accuracy. If you're interested, we have a QR code. It's not available at the moment, so it's very much register your interest and we'll get in touch. Again, so we will be talking about methylation and hydroxymethylation during the external speaker presentation. There is no additional library preparation required. There is no treatment of the DNA required. You will produce a most complete human methylome with nanopore data. I think with bisulfate, at the moment, you get about 85% of the CPG, you get 98% of the CPG, and the non-CPG methylation in the genome. We have I would recommend reading the excellent paper from Kolmakov. They are working with the NIH Scott in the US on Alzheimer's disease and also other sort of neurodegenerative disease. They have really developed solutions to enable methylation call at scale that are highly concordant with either bisulfate sequencing and the data they had with EPIC. And again, it's really easy. As you can see with the toggle, just switch it on. So this is our instrument. I don't really like the fact that we put benchtop in the middle because everything can be a benchtop instrument. Everything does fit on a bench. But we have our more distributed sequencer, and I think everybody has heard or has seen a MinION, and we celebrated its 10 years last week. We have mid-size benchtop sequencer. I really encourage you to come on the booth to see the P2 integrated. It's really, really cool. And then we have our more high throughput instrument. When it comes to looking at data in particular for rare disease, we do realize that the clinical genetic lab are really busy and they do need support for end-to-end interpretation of the data. And we're working with 4 different companies at the moment for variant prioritization and interpretation. They are Fabric Genomics, GenX, Sequence Genomics, and Varsa. So if there's 3 things I would like you to remember is that with Nanopore, you'll get much richer insight. You're getting very accurate genomic data for all types of variation. You can get much, much faster result than with any other technology. Near sample real-time workflow, don't need to batch, and it's really accessible, it's really affordable. We had lots of examples during the conference of people saying, "We've embraced Nanopore because it makes it accessible and very equitable."
Thank you very much for those insights, Cora. We'll now hand over to Greg.
Thank you very much, Nanopore, for inviting me. It's very kind of them. I always surprise when Nanopore invite me to talk because they know that I'm going to hang out the washroom, but we'll see how it goes. Okay. So these are benchtop PromethIONs. These are in the lab. We have 3 of these, and that's what we're working with at the moment. And I'm going to talk to you a little bit today about how we're trying to transform what, when I started working with Nanopore, was essentially a research platform into a clinical platform, something that's robust, reliable, and can work at pace for both cancer and for rare disease. So a very quick intro, if you don't know anything about Genomics England, they were a project that was set up by the UK government in about 2012, 2013. The aim was to look at the feasibility of introducing whole genome sequencing as a standard of care test in the NHS, in the National Health Service. So that project started slowly but ramped up quite rapidly. And in the years between 2016 and 2018, about 85,000 of those genomes were sequenced. And in fact, in 2018, we sequenced over 50,000 whole genomes. So those are real people's genomes. They're not reference genomes. They're not gold standard or platinum genomes. And so what we had to do is establish quite a significant pathway from patient or participant through to interpretation and returning results. And so that's what this is all about. It's about working with long-read sequencing, with nanopore sequencing, and trying to essentially replicate that pathway, bring in all the benefits of nanopore sequencing. So over the last 3 years or so, we've been working on some projects in the R&D lab that was established about 4 or 5 years ago to assess new technologies. And we have been working almost exclusively with Nanopore, despite the fact I might tell you of some of the issues and difficulties we've had. I absolutely think that that is the way to go. So this is the team at the moment that essentially does the sequencing and the QC and the processing and the early analysis. There are lots of other teams involved because in order to develop an end-to-end pathway, we need to have a lot of cooperation between different teams and also with stakeholders, with the NHS and with the genomic laboratory hubs that lie within the NHS. And I'll tell you a little bit about that in a bit. So we have a pretty straightforward workflow. It's made slightly more complicated by the fact that the Research and Development Lab at Genomics England actually is embedded within the Sanger Institute. They very kindly host us. So we have built an infrastructure that allows us to analyze and process data off machine. So we don't do it on the boxes on the machines. We actually have a 10 gigabit link to a cluster of NVIDIA boxes where we do our processing. So very recently, we had quite a lot of strain on the... I did have a pointer here. Yeah, here we go. So we had quite a lot of strain on the this Weka storage here. So in order to alleviate that, we are now sending all our data up to AWS, and we only call it down to be processed when we need to. So that has actually removed a bottleneck, and we're just in the process of implementing that at the moment. So this is a slide you'll see a couple of times in the talk. It's just a summary, really, of what we've been doing in the R&D lab over the last 4 years or so. We've worked on a whole heap of different kinds of samples. Some of those samples are fresh. We've extracted them ourselves from blood or from tissue. Some of those samples are 20 years old, archived samples that have been sitting around in freezers. So there is a real mixture of samples. And that's a really important point to note here, because it's all very well showing data from reference samples that are beautifully extracted from tissue culture, but this is real data from real samples that have been extracted in a host of different ways. So I will come back to this. So our kind of journey is summarized in these rather bare plots, but each individual tiny circle on these plots is essentially a flow cell. And for us, a flow cell is essentially a kind of 20 to 30x genome. So you can proxy those dots as genomes. And what I'm trying to show here is that there's been a tremendous development over the last 4 or 5 years. And when we started working with Nanopore, they definitely weren't clinical ready. There's no doubt about that. but I think we are now at a real tipping point where that's changed. And that's changed because of a number of different advances in different ways. So highlighted here are a couple of updates of versions of Guppy. There have been lots of them, as people who work with Nanopore know. Sometimes that can be challenging in itself when you're sequencing a large cohort. But each time we get a significant update, we've tended to see a bit of an uptick in the quality of the sequencing we generate. We also, of course, have moved through different flow cells. So the R10 has definitely shown an uptick over R9, and I'm sure that's been universally reflected in virtually every application. And moving to 5 kilohertz more recently has also shown a significant uptick. But as you can see, there has definitely been quite a lot of progress over the last few years. Sorry. Okay. That's quite scary. So what I have here is another way of summarizing those data sets, and we've split them up here into cancer and non-cancer. The top panel there is the top panel here is showing you the sequencing depth. That has been quite variable, and the reason for that variability is one of the things that we find constantly challenging. Quite often, it's the quality of the sample we're working with, the way in which we treat the sample. Sometimes, it has to be said, it's the flow cell quality. And so teasing those two apart and getting a real consistent quality of depth is quite challenging at times. I think it's getting better, but not as quickly as we'd like it to. We've managed to surprisingly keep a fairly consistent N50. So we've tried to do that. We're trying to find that kind of optimal balance between coverage and the N50 that we generate. And for most samples, we managed to do that around the kind of 15 to 20 kb mark. For some sets of samples, so there have been some cancer samples where we've really struggled with that because the quality of the DNA has actually been quite poor at input level. And then the most obvious one, which is reflected in what I was showing earlier, is that the actual read identity has got much better as we've worked with this, and it's reached a level now where we're confident that we can call pretty much everything that other sequencing technologies can call and more. So I just take you back to this slide. The last 3 years, the reason why we sequenced a lot of those samples, not all of them, but a lot of them, is because we've been involved in some really key initiatives, which are part of the kind of Genome UK implementation plan. And so I'm going to talk a little bit about the Cancer 2.0 program, and then I will mention a kind of rare disease program that we're also working on at the moment. So the Cancer 2.0 program has its own lead and its own team. Emma McCargo is the lead. And over the last 3 years, and we've just moved into the final year of the 3-year cycle, the aim and objective of that project is to take nanopore sequencing, as I say, into the clinical space, and ultimately to get it to a stage where it is a standard of care test in the kind of test directory that the NHS has. So in order to do that, we have certain objectives, and these are quite key because they differentiate this approach from other sequencing and other test approaches. So we want to look at faster diagnosis. At the moment, the way in which the end-to-end pipeline works for whole genome sequencing, it's all based on Illumina sequencing, and it means that there are certain operational approaches that make that limiting in terms of how fast we can make it. So we'd like to use the versatility of the platforms with Nanopore to speed that up. And in order to do that, we're distributing the sequencing close to the patient, so in the first instance to the genomic laboratory hubs, and there are 7 of those around the UK. So obviously, and reflecting the previous talk, we want to have a more comprehensive genome. Sometimes we might want to have a tailored genome. We might want to look at specific features rather than all of them. But the idea is that all these tests can be run on Nanopore platforms. We have to, as part of our due diligence and as part of our the fact that we are essentially a government organisation, constantly review which sequence technology might be best placed to do this work. And we have a number of fundamental reasons why we think that Nanopore is best placed to do this at the moment. And the final one is about evidence generation. So one of the objectives was to generate sufficient evidence to show that we can replicate what short-read sequencing is providing and also provide added benefit as well. So we are doing some quite specific analysis across specific cancer types. I'll go into this in a little bit of detail, but because we're a bit limited on time, I'll probably move quite quickly over some of the details. So by all means, kind of talk to me about it afterwards or contact me via email, and we can give you some more in-depth information on this. But we're looking for to be able to pick up the kinds of variants that we pick up with the existing tests, the existing standard of care tests, with the ultimate aim potentially of replacing the test, but at the moment just to look that we can cover a number of different tests in the genetic test directory. We want to make sure that it works well in a clinical setting. So that is largely around what I mentioned earlier, that quite often there are a number of different extraction techniques, particularly in cancer where you're extracting from solid tissue. You get quite a number of variables around the kind of tissue, the kind of extraction you're doing, the way in which you resuspend. There are many things that actually have quite a significant impact on the quality and the amount of DNA that you get. So we want to make sure that is good enough and that we have the right extraction methods for that. We want to increase turnaround times, and there's no reason why we can't do that, we think, especially with the versatility of the platform, being able to just run one flow cell at a time, potentially. And that links to the right cost as well. Obviously, with our sequencing set up at the moment, we sequence a lot of genomes through Illumina, but we have to make sure we fill flow cells to be able to continue that kind of throughput. So this, again, is more versatile. So taking all that into account, we've been doing quite a lot of variant analysis on the data that we're generating on those cancer types I showed earlier. When I put this image in, I meant it to reflect variant analysis and not the cancer bioinformatics team, so apologies if that comes across slightly strangely. So going back to this slide, as you can see, the cancers that we focused on largely are the cancers that are already in the test directory for whole genome sequencing with short reads in the NHS. So that's the brain cancers, hematological cancers, and sarcomas. And we also try to focus to a degree on pediatric cancers as well. So these are the samples that we have used to generate the evidence. These are samples from the 100K participants. So they are, and I use the word very carefully, but they are essentially leftover DNA samples that we biobanked at the time when we did the short read sequencing. So that means they're quite old. They were extracted with no view generating high quality or large fragment DNA. They were simply extracted for Illumina sequencing. So we have had some limitations on the quality of data that we can generate because of that. But despite that, we were very keen to look at concordance and to see if we can actually pick up all the variants that short read does. So there's been significant work from the cancer bioinformatics team led by Ilona Sosinski. And again, I'm not going to go into this in detail, but we've been looking at runtime because obviously we're trying to do high throughput and quick turnaround times. And we're looking for recall and also false positive rates. So if you focus on the greens, there are a couple of callers that we're really happy with at the moment. And we're working with both of these callers in different contexts. And in particular, there's been some benchmarking of Severus performance against some specific structural variants that were called, or in some cases, and there were 4 of these, that were not called in short reads, but may have clinical impact. So whilst we are picking out all of the structural variants that short reads identify and other tests, other genetic tests in the NHS, we're also picking out additional ones. So why did short reads miss them? Well, because of the complexities of genomic sequence and the fact they fall into or adjacent to repetitive elements. CNV calling we thought would be quite simple, but in fact it's ended up being quite complicated, and we've had to exclude quite a few CNV callers because they just don't do the job, but there are lots of CNV callers out there. We're collaborating with a number of groups on certain CNV callers, the same way we're collaborating on structural variant callers. I've been informed recently that a tool called Hatchet2 is working really well, so that's one that we're hoping to implement into the pipeline very soon, and it seems to successfully pull out CNVs that are pulled out using other methods. So we're also looking at methylation. I won't talk much about methylation because I think you'll hear quite a lot about methylation in the next talk. But just suffice to say, we've been doing the same kind of benchmarking work. It turns out it's no surprise that methylation calling is very good, and the classifiers that we can compare with all run very well using Nanopore data. So in terms of where we are at the moment, we have nearly built an end-to-end kind of bioinformatic workflow now for the clinical pipeline that we're hoping to deliver for cancer. There's a couple of areas that we still need to do a bit of work on, CNVs and the methylation classification. And what that means is that it adds quite a level of complexity to the way in which we're running our pipelines, and this is something that should not be underestimated. When we're trying to build new pipelines using large quantities of data and doing processing that still takes a significant amount of time, there's still a significant amount of GPU and CPU that's required for a lot of the analyses. So we're currently trying to separate the two pipelines. This pipeline in blue is what we hope will end up being a pipeline that we can look to try and accredit so that we can actually start to push for clinical adoption. But we will still retain an R&D pipeline which will allow to do further development. So these kind of things do take quite a lot of additional support from IT, infrastructure, other groups. So that's a large part of the organisation at Genomics England. So we're in the last phase now. Last year, we started working with 2 genomic laboratory hubs, 2 out of the 7. We call them Pathfinder labs. That work has been going very well. They have started sequencing their own cancer samples, and we are looking at the analysis of those using the newly constructed pipelines that we're building. This year we will be scaling up and hopefully rolling out ultimately to all 7 GLHs to cover that kind of equitable access that we have to provide for the NHS. So very quickly, in the last couple of minutes, 3 minutes, I just wanted to talk about what we're doing with rare disease. So rare disease has always been something that Genomics England has focused very hard on. We have obviously we've done about 85,000 whole genomes associated with rare disease trios. So that's about 28,000 trios. So the diagnosis rate from those is around 25%, and we can quibble about 5% either side, but it's still nowhere near what we would like it to be. So we've already sequenced quite a few samples from the 100K project. These are samples from the 100K. There are some others here that we've done in individual projects. So we've done about 1,500 individuals from the 100K project with long reads now. But this year, we're hoping to expand that considerably, which is why we have such optimistic figures here. We're hoping to launch the equivalent of the Cancer 2.0 project as a Rare Disease 2.0. And that is part of a multi-omics project that we're running, where we're also doing transcriptomics and metabolomics and proteomics. So this is a partnership that we're building between Nanopore, Genomics England and Eurofins, based at Milton Keynes. They're building their Nanopore infrastructure so that we can put high volumes of samples through using a standard protocol. So that fits into our overall project, as I say, where we're hoping to combine data from a number of different modalities to optimize diagnostic yield. We have a process flow that which is almost completely developed now. We're going through the early pilots to make sure that is running smoothly, and that also includes, amongst that, at Genomics England, using the Nanopore epitome workflow. So what do we want to get out of the RD 2.0? Well, initially we want to increase the diagnostic yield for the 100K participants, and that's linking it with the rest of the multi-omics that we're doing. We also try to provide a comprehensive data set for researchers. So we have a research network associated with Genomics England who can access the data. And we are also trying to build higher throughput protocols for running Nanopore at speed. So at that point, I think I'll finish. I think it's been quite, I wouldn't call it a rocky road, but I think it's been a road where we've had a lot of challenges. We still have some challenges, and moving from a research platform to a clinical platform is still a major challenge for Nanopore. We're working with essentially a living sequencing technology as opposed to a chemically derived one. So I think those challenges will remain, but I think we can harness those sufficiently to be able to generate really good clinical tests in the future.
Thank you very much, Greg, for a great presentation. And now, finally, we'll hand over to Brynja.
So thank you for the introduction, and thank you for inviting me to speak here. It's a great honor to tell you a little bit about the large-scale methylation studies that we are doing at deCODE using Oxford Nanopore sequencing. For full disclosure, I'm an employee at deCODE, like mentioned, and it's a subsidiary of Amgen. I think you're all now pretty much aware of what the ONT, how the sequencing is working. So it's they're monitoring the electrical signal while they pass the DNA through the protein nanopore. And then it's base calling algorithms that take care of decoding this signal into DNA sequences, the bases. But for the methylation calling, the early days algorithms were basically a second step on top of this base calling algorithms. So you needed an additional algorithm for the modified base calling, like the methylation calling. And that was, for example, Nanopolish was one of the first algorithms that did that efficiently. But now you're actually having the methylation step, methylation calling, incorporated it into this base calling algorithm. So it's just the base calling algorithm is trained using this expanded expanded alphabet. So it just add, in this case, the 5-methyl-C to the alphabet and train the modified base caller on that. So this is, I'm talking about mostly the 5-methyl-CpG methylation, but you also have other type of DNA modification that you can detect using similar steps. So at deCODE, this data that I'm going to present today is mostly based on this 7,200 whole blood samples, and they were sequenced on 9,000 Promethean flow cells, and they were sequenced to a roughly about mean of 17x coverage. And this is because also we started the sequencing in 2018, and a lot of the sequencing was done around 2020. So it's a lot of early days data as well, but this has definitely been increasing over the time. So yeah, we have them all methylation sequenced using Nanopolish, and again, that's just because early days that Nanopolish was the way to go. And now we have also started using the guppy for the guppy, or now even more recently in Dorado. Thank you. So yeah, just because we started this in the early days, we were going with nanopolish, and that's why all the samples were methylation called using nanopolish. But also this is already a study that we did like 1.5 or 2 years ago. So we only had now, at that time, 300 samples that were additionally methylation called using guppy. But now we have already methylation called all these samples. And we also have now starting to use Dorado in more scale. And this is also mostly presented for R9 flow cells, but we also have some samples for R10 flow cells, and it's really nice to see how the data has changed over the time, and R10 is really showing improvements. But for validation, we also have 130 samples that we had sequenced using oxbisulfite sequencing, and we kind of treat that as our ground truth, just to start with. So we look at the 132 samples that we have sequenced both using Nanopore and the oxbisulfite, and it's from the same blood draw, and we can see a really good consistency between the two methods. So the overall methylation level per samples, the difference between the two means is less than 1%. And we can also see that the correlation is really, the Pearson correlation between the two methods is extremely high, and it's about over 0.8 for the Pearson correlation coefficient for all samples almost. And especially if you look at 20 to 30x or higher, you can see that the correlation is very good. So we definitely see that there is consistency between the two methods. And I think this was very important because when we were starting to write our first paper using the methylation data, this was what we had a lot of questions from the reviewers. They were really asking us to validate the data because it's a new data and you need to show that it's actually working as we expect it to be working. So also we looked at just some known methylation qualities, like we know this bimodal distribution, and we see it in all 4 data sets. So using nanopolish, also using guppy, and especially for the new flow cells, we can see that the early days guppy data had the peaks were a little bit shifted towards the middle, but now in the new data shown in the pink, like for the new flow cells and newer versions of guppy, this is, for example, solved. So it's showing the improvement over time. We also can see that we have more CpGs that detected than in short-read sequencing. And here it's actually shown like nanopolish is detecting fewer, but it's more based on the criteria that you have for each algorithm. And also we see that the lack of methylation within transcriptional start site is seen for all 4 methods. And we also see that this behavior is mirrored here, that the methylation is it's not going as low for the unmethylated regions as for the R9 flow cells and the earlier versions of guppy, but this is also solved for the newer versions of the data. So due to time reasons, I'm not going to go into too much detail, but we did talk about we did some filtering, especially on the early days data. We were seeing, for example, strand bias in the data, and this was even more for nanopolish than for guppy. So definitely it's less of a problem with more recent data, and it's much less CpGs that we were filtering out for the newer version of the data. But you can definitely look into doing some kind of creating some kind of high quality set of CpGs by using these methods. And this is all part of a paper that we published in Genome Biology in March. And you can definitely go there and look more into and see what we did there. But so just to summarize what we found out in that paper, it was that CpG methylation calling of ONT data is highly consistent with oxbisulfite sequencing, and you can use quality filters to further enhance the accuracy while removing at most 30% of the CpGs. And this is at most because for newer versions of data, it's much less than that we were removing. And also, improvement in the base calling algorithm and the flow cell chemistry resulted in lower error rates, reduced strand bias, and improved methylation calling. So this was basically written as a sister paper to this paper because we got the first round of reviews were really digging into how we could validate the data. So we decided to just write a paper, and in the future we can always reference back to that paper. And this is a paper that has now been accepted in Nature Genetics, and it's a project led by Ólafur Stefánsson at deCODE. We have kind of formed now a team. I'm more on the bioinformatics side. he's more on the biology side, and it's very good to have a team of those working together when you're working with big data, but also trying to understand what the data is telling you. But yeah, this should be published now in the next month or so. But just a short introduction about what we wanted to do in this paper. So genomic regions where the adjacent CpGs are unmethylated often corresponds to regions bound by transcription factor. So these regulatory regions, methylation of those regulatory regions is widely considered to affect gene expression. So our aim was to understand the contribution of the sequence variants to CpG methylation and determine the relevance of CpG methylation to gene expression in the context of the variants. So first, what we have to do was to phase the methylation. So this is where long reads really show their strengths. It's much more accurate phasing that we can do. But the way we did it is that we took advantage of 34 million SNPs that had already been imputed into all of our individuals and used them to assign the parental haplotypes to the long reads. So we define methylation-depleted sequences as having 3 or more consecutive CpGs unmethylated on at least one haplotype with no more than 500 base pairs apart. So the reason why we do the phasing is so we can be working with the variant and the methylation at the same strand level. Because we're only working with cis in this paper, it's very important that we can do it at the same strand level and have the exact CpG ratio at this strand level. But this way, we identified about 190,000 depleted sequencing, and we are both capturing known regulatory elements and also finding something novel, like something rare events that might be very important in gene regulation. So next, what we wanted to do is just see how the sequence variants correlate with CpG methylation of those depleted sequences. So we used a set of 34 million high-quality sequence variants, and we looked for association between the methylation and the variant within 100 kilobases. And we used just linear regression to test for the association, and we also adjusted for gender and age, and also some principal components. So this way we found that about 40% associated with a sequence variant. And we refer to these variants as allele-specific methylation QTLs, or ASM QTLs in short. And we also saw that by looking up in the we saw that these were enriched among these that were known to be corresponding to transcription starting sites. So yeah, so next we wanted to see if the variants correlates with mRNA expression. So we had 900 samples that were sequenced using, that are RNA sequenced as well. So we phased the RNA sequences, and then we looked for association between the RNA isoforms and the methylation. And we found that the methylation-depleted sequences were, out of those 80,000 that were found within 100 kilobases up or downstreams of the transcription start site of the mRNA isoforms, there was 1% that associated with mRNA isoform. So, and thereof, about 4% of those were associated with the MDS correlate with a transcription start site of an associated mRNA isoform. So, but interestingly, we found that ASM QTLs dominate in correlations found between MDS and mRNA expression. So because all of the variants that associated with, all of the methylation-depleted sequences that associated with mRNA expression also associated with sequence variants. So as you see here on this figure, you might think this is due to different level of variants, but we show that it's nothing to do with the variants. Additionally, you can see here that the variants explained by the mRNA, the variants in the expression is explained more by ASM QTLs than the methylation. And even when you adjust for the variants, the methylation, there's no residual correlations left between the methylation and the variants. No, the gene expression, sorry. So this shows that the correlation between CpG methylation and gene expression is largely driven by sequence variants. So next, what we wanted to do is to see if the ASM QTLs were enriched among the GWAS signals. So we looked for the high quality variants and association between about 260 diverse human traits. And what we found was actually that the enrichment was on average 23.2-fold. So the ASM-QTLs were 23.2-fold enriched among the GWAS signals. And it was more for hematological traits, which is not surprising at all, as these are, as methylation is tissue specific, and we are working with blood samples here. But it was still, for other traits, it was 6.6-fold enriched on average, and it was actually only outshone by protein coding variants, with missense variants of 98.7-fold and loss of function variants of 292-fold. So we actually, this overrepresentation in the GWAS catalog shows that these are very important variants, and that we can actually see here a few examples where we have them in strong LD with known disease variants. And for example, with example 1, we have this variant that is associated with type 1 diabetes in strong LD with ASM-QTL, found in association with the promoter methylation of this gene here. So here it's actually the disease-associating variant is not in this gene, but we are actually using the ASM-QTL to point us towards the potential target gene, candidate target gene. And in example 2, we have a variant associating with asthma in strong LD with ASM-QTL that associate with methylation rates of MDS within enhancer element intronic of this gene here. And here it's actually pinpointing just very narrow region of importance in relation to this disease. So these ASM-QTLs are definitely showing us where in the genome there might be important regions for the disease. So just to summarize quickly, we investigated correlation between CpG methylation, gene expression, and alleles of sequence variants on a haplotype level. And these results support the notion that ASM-QTLs influence methylation of CpGs through their influences on protein binding to DNA. We also show that ASM-QTLs are relative to human traits, and their influences appear to be tissue-specific. And our important part is that our results suggest that the sequence variants is the primary driver of correlations between CpG methylation and gene expression. So lastly, I just want to use the last few minutes to talk to you about our future work at deCODE. So one of our main first projects will be regarding aging. So it's known that the methylation is a good predictor of age, for example. There are two mainstream clocks of Horvath and Hannum, both clocks that you can just use very few CpGs to predict age. And we have actually seen that this is not working very well in ONT data, or at least not in our data set. So we have created new clocks for that. And we actually just want to try to understand the aging better. We also have some proteomics data available at deCODE, and it has also been shown that you can use proteomics to predict age and use some sort of understanding the disease, how aging affects diseases, or the other way around. So we really want to dig into that and try to also now we have all this data. We have the genomic sequencing, we have the RNA, we have the methylation, we have the proteomics, and now we really want to find ways of connecting it and try to understand. And I think aging is a good way to start, like how do proteomics affect the aging, how do methylation, and can we even make better predictors of age and disease progression using these two data sets together. And I think also, next point, I mean, everyone who went to the clinical sequencing slot or the long-read sequencing in the clinic yesterday, I think we know that this is a very good potential with ONT to use this for improve the solving rate. And we actually had a really cool case a couple of months ago where we had identical twins coming to us. One of the twin was severely affected with some very severe symptoms. And actually we had to, our solution was just it's inconclusive. We don't know what's wrong with them. And it's more of we're just like supporting the hospital, but the hospital in Iceland then sent it abroad for some clinic where it's actually very specialized in solving these kind of cases. And only based on difference in the methylation pattern, he was diagnosed with Engelmann syndrome. So this is something that we were okay, the kid now has the solution, but we still just wanted to see if we can see it in the ONT data. And this is something that we could have been able to solve if, now we knew what we were looking for, so it was easy. But this is something that we can definitely use to help improve our solving rates. And then it's for clinical biomarkers. I think it's very good potential to create some kind of clinical biomarkers so we can detect diseases earlier. I think one very good example would be clonal hematopoiesis. That would be a really, I think, a low-hanging fruit to look at that if we can create clinical biomarkers with methylation for that. And lastly, I think it's a really good predictor for phenotypes. Like, there are a lot of phenotypes that are self-reported. For example, if we think about smoking, it's really dependent on how the question is asked and how you are willing to answer, how the phenotype will be created. So by using because we know that smoking affects the methylation patterns of individuals, we can use the methylation pattern as a predictor for smoking behavior. So I think that's one example of how we can use this data in future. We also are really excited at looking at the Y chromosome. I don't think a lot of methylation studies have been done there, so that's another potential. And same with the X as well. But yeah, I think that's it for me. I just want to thank all my colleagues at deCODE, especially Ólafur Andri, who has done a lot of the work with me, and also our colleague at Amgen and the Nanopore team for supporting us through the year.
Thank you all very much. I'm sure you'll all agree that there have been some excellent points raised during the presentation. Just to mention that there will be a short survey at the end of the webinar, and we'd really appreciate it if you could just take a moment to complete that. So we'll now begin the Q&A session, for which I'm really pleased to say we've been joined by Melanie Tango, sequencing R&D lead at Genomics England, who works with Greg in Greg's team. So she's joining us on Greg's behalf for the Q&A. To submit a question to our speakers, you should enter your question into the box on the right-hand side of the screen and click send, and we'll answer as many as possible during the time available. So we've already had a few come in, so I'm going to dive straight into the first one. What do you anticipate the impact is of including methylation in your dataset on diagnostic yield?
Is it the question for me or for Melanie? Sorry.
I think this one's probably directed towards Melanie.
Yeah, okay.
Sorry, could you repeat the question?
Sure, no problem. What do you anticipate the impact is of including methylation in your dataset on diagnostic yield?
Right, so I think we haven't really started to look at like the role of methylation in cancer, which is our program where we're in cancer, we are definitely looking at methylation, but we are looking at methylation mostly from the classification perspective, especially in brain tumor. Methylation in rare disease is something we haven't explored yet. And I think as everyone may be aware, this is where really there might be a need to increase our diagnostic yield. So the true answer is I don't know yet, but certainly something that would be interesting to investigate.
Fantastic, thank you very much. I think this one's directed towards Brynja. Can you comment on how the work at deCODE is progressing with reference to the latest publication?
Yeah, so now we have added this ASM-QTLs to our set of variants that we look at as variants of interest. Not a lot of hits yet that we have been using this for, but it's definitely something now we added to our catalog of variants that we are looking at as potential disease-causing variants. They've also helped us like pinpoint down these regions of interest where we can actually find the exact location in the genome where there might be of interest in diseases. We've also been trying to use these variants to understand how maybe the environment is affecting some of the diseases. Some of the diseases are known to cause, for example, let's say lung cancer, and then we can use the methylation and the variants to try to understand if the environment is like smoking is very affecting in lung cancer. So we're trying to understand the environments as well by using these methylation related variants.
Excellent, thank you very much. Next question, I think this one's directed towards Cora. What are the data storage needs necessary to set up Nanopore sequencing to include methylation calling for larger sample numbers?
No, I think it was for Melanie actually. It's specifically what did you
Oh, apologies.
at Gel.
It's 12 and they are testing the fire alarm every week at 12. I had forgotten. Could you repeat? It was very loud.
Sorry. What are the data storage needs necessary to set up nanopore sequencing to include methylation calling for larger sample numbers?
Right. This is a fair question. Large, I think, is a word that would describe it well. So now the real question that we need to answer, in order to make the technology viable for the healthcare system is what do we need to store? So obviously there are several type of file you can store. If you want to store your raw signal, I would say it's not viable for the healthcare system, just simply. It's just too large. At the moment, we still do, because we are still, for us, we are still in an active research stage as well as implementing the clinical stage. So there is still an interest, but that's really something that we will consider we will reassess. We are constantly assessing the need of storing these extremely large, POD 5, FAST 5, depending what you're using. Now, if you are talking about storage of BAM files, you're already kind of reducing your need. That might be one option. And if the technology stabilizes enough, and you are only dealing with your VCF files, this is obviously a different questions. You have to realize that in the healthcare system in England, so for the NHS, whole genome sequencing with short read sequencing is standard of care as of now, and we are also dealing with the same problem. So the specificity of long read technology is really about the source file with all the rich signal from which you can extract lots of potentially new information. That seems quite a stretch to say that it will be viable for any healthcare system.
Brilliant. Thank you very much. Next question. Are you planning to look at both 5MC and 5HMC in the future? I'm not sure quite who that one's directed to.
I'll reply quickly before. Yes.
Yeah, and same for us. It's definitely a future project for us. Right now, we are trying to find, or we are actually just working on getting the equipment needed to kind of be able to validate the data until we want to use it. We want to be able to validate it as we did for the methylation before. Also, it's more relevant when you have some other tissues than blood. So as soon as we are examining maybe our other tissues samples, that's going to be of much more interest, especially if you have some brain samples or something like that. It will be of a high interest for us.
Excellent. Thank you both for that. This one's directed towards Brynja. You mentioned about characterization of allele-specific methylation and the impact on gene expression. Could this have been possible without phasing of the alleles with long reads?
No, for this analysis, the phasing is a key component, because that way you're definitely looking at the variant and the methylation at the same strand level. So it's definitely very important that we are able to phase the data, and phasing of the short reads sequencing is much less accurate, and because we're using those SNPs, you need to have a SNP within the read. So by having the longer reads, it's much more likely that you will have a accurate or a SNP that we can. And also there are other methods to phase it, and it's definitely more accurate in the long read sequencing data. So that's definitely a key. We would not be able to see it without phasing.
Fantastic. Thank you very much. This one's for Melanie. As part of the Cancer 2.0 and Rare Disease 2.0 programs, you have chosen to include long read sequencing technology. What were the determining factors that influenced your decision to include ONT specifically as the long read technology of choice?
Okay, so I maybe the answer is slightly different, like for both programs. So RD 2.0, so rare disease, like undiagnosed cases, is like much more of a research program than the Cancer 2.0, which is definitely like towards the implementation of like this technology in the clinic. So in terms of Cancer 2.0, we looked at like the main technologies currently on the market, and in terms of scalability and in terms of like distributing the model of sequencing, if I may say, sometimes just in terms of footprint, Oxford Nanopore Technologies seemed like the right way for us to progress. Really like having the opportunity of like developing pipeline, knowing that you will be able to do the sequencing on a tiny machine or a very big high throughput machine is quite important for us. For RD 2.0, as I think it was explained like in one of the slides, we are looking at this from a very multi-approach perspective. So we have used different types of long-read technology. We are maybe not running them internally, but we have some PacBio data sets, we have some ONT data sets, and we would be happy to explore other type of omic approach, regardless of the technology they are coming from.
Excellent, thank you very much. Next one for Brynja. What is the impact on new mechanisms of disease? You identified 34 variants in transcription start sites in blood and in association with type 2 diabetes and asthma. How do you see this with other tissue-specific disorders?
So we have mainly been examining blood until this point. So it's mostly blood-related diseases that we're seeing the hits on. This is definitely something that we believe would be seen also in other tissues. We at deCODE do not have a lot of tissue samples, so it's something that we will not be able to investigate as well as we would want to. But I believe it would be the same. It could give us important information about tissue-specific diseases as well if you're examining that tissue. So if you would be looking at brain tissue, you would maybe get more insight into neurological disorders. But unfortunately, it's not something we can do at this point at deCODE.
Brilliant, thank you very much. And next question. Is there any use of AI in Nanopore?
Is it for me or anyone?
Whoever would like to jump in, please do.
Yeah, I mean, definitely the base colors and the methylation colors are based on AI, so they are definitely using it. And also for the analysis, when it's this big data, you're always using some algorithms that you can call AI, I guess.
Yes, I was going to jump in on this one. I guess it depends what you mean by AI, but none of the analysis would be accessible or possible without the use of algorithm and some form of automated scanning of the data. It's as both Brynja and Melanie have explained. It's a very, very data rich.
I think we have like to. This is something as scientists maybe we need to be very careful about, or careful or just spending a bit of more time doing some educational piece here. We are working with not only like patient but like mostly research participants. You know, the main principle of like you signing a consent to do research is to understand what you are signing for. So I think it's really our duty to talk a lot more about like what we are calling AI, what are the risks, what are the promises, what you are exactly going to do with this technology. I think it's a very wide topic that is not the topic of today, but it's an interesting question.
Excellent, thank you all. We've got time for just one last question. I think this one's for Melanie. Are you able to comment on the equity of access across the NHS of longer read technologies and the benefits they bring to the patient?
Okay, so I think it's an interesting one. It's an essential factor. I think we are, or I don't know for other country, but the few countries I follow, I think equity of access of like new technology, especially in healthcare, is absolutely horrendous. Like in any of our system, and will long-read technology fix all of that? Certainly not. But I really think that if we are pushing this new technology and like maybe multiplying like the number of approach that we can bring to the patient to help their care, this is like how we are going to maybe like bridge a little bit of the gap. So it will not fix everything. No, I'm not saying that, but we are really working actively on the Concert 2.0 program to put sequencer as close from the patient as we can, with the sequencing step, the sample collection steps being as low key as possible. And I think this is what will bring like the best outcome of new technology to anyone, anywhere. That's what they say, right?
Brilliant. Thank you very much, Melanie. So unfortunately, that's all the time we have for questions today. Thank you all so much. I'm now briefly going to hand over to Cora just to mention some upcoming events before we close out.
Thank you very much, Karen. I would like to thank again our speakers. Thank you, Brina. Thank you, Melanie, for jumping in and covering for Greg, who couldn't join us today. So I just wanted to cover a couple of things. The summer is over, so we are now starting again our What You're Missing Matter tour. And as you can see, we've got a number of dates in Europe and in the US. This is the second part. We had a number of dates earlier in the year. So if you don't see your favorite city on there, it might be because we have done it earlier in the year, or we will do it most likely coming after those dates there. And I think that's it for
Perfect.
today.
Thank you very much, Cora. So that just leaves me to say a huge thank you to everyone, for our presenters and for our audience. Once again, thank you all.